Welcome everyone to this Connect Science seminar today. And while everyone is still trickling in, it's my distinct pleasure to introduce Kevan Shokat to this audience, who I'm sure does not really need an introduction here at Dana-Farber. Kevan joins us from University of California in San Francisco, where he is a professor in cellular and molecular pharmacology and also in chemistry. And he's also an investigator of the Howard Hughes Medical Institutes. Before coming to San Francisco, he received his undergrad training in chemistry from Reed College and then joined the back then still early Pete Schultz for his graduate work at UC Berkeley, and followed this with a short postdoc at Stanford before then becoming an independent investigator at Princeton and ultimately moving back to the West Coast. I think all of you know that Kevan's work has really been groundbreaking at many levels for the field of chemical biology, starting with the development of tools that now everyone uses, such as engineered kinase ATP analog pairs to elucidate the biological function of kinases, and more recently, really groundbreaking discoveries in the space of innovative medicines, such as efforts in cracking the targeting of KRAS, that I'm sure he's going to be talking a little bit about, and going on from this in many different directions. He's won numerous prestigious awards along his career, and just recently, he's been Alfred Beta Award in bioorganic chemistry, and he's also won the Jonathan Kraft Prize for Excellence in Cancer Research. I don't want to take much more time away, so let me just say one more housekeeping item, which is that if you have questions, please post them to the Q&A tool, and we'll get to those at the end. We'll read them out, and if there are any left, we'll make sure we send them to Kevan. Kevan, it's my real pleasure to have you, and please take it away.
Thank you, Eric. Thank you for inviting me, and I thank all of the Dana-Farber faculty and staff for putting these this session together. I looked up the previous ones and downloaded and watched and spent a few hours last night. So it's a great resource, and it's great to see everybody come together during the pandemic and create new ways to share science. So looking forward to this. As Eric said, I'm going to talk about ways to inhibit RAS-driven tumors. And I'll start with small molecules that target one of the KRAS alleles, and then try to go on to explain how I think, now that we're probably 8-10 years into the immuno-oncology revolution, how maybe we can leverage chemical direct targeted therapies and merge them with immuno-oncology. And then at the end, I'll try to tell you about a new direction in the lab, where we're, instead of going after oncogenes, we're trying to elicit responses in cells that are akin or directly related to tumor suppressive activities. So before I start, I'll tell you that I've got a number of disclosures of companies that I consult with or have emerged from compounds that we discovered in the lab. And so they're all here. And I'll mention that probably the most relevant is from Araxes, Wellspring, and Janssen, and RevMed, and Erasca. So we're all aware that mutations in proto-oncogenes and tumor suppressors drive cancer. And KRAS is the most frequently mutated oncogene on the driver's side. And on the tumor suppressor side, P53 is the most frequently mutated tumor suppressor. It's also the most frequently mutated gene of all. Something like 50% of tumors have some aberration in the P53 gene. So to put that in perspective, we think there are about 18 million new cancer patients a year in the world, and 2.5 million every year will emerge with a KRAS mutation, and about 9 million will emerge with a P53 mutation. So this is a very large slice of the pie of cancer tumors that emerge. And so we've been working on KRAS for a number of years, and I'll take you through those efforts. We know also that KRAS and P53 are a somewhat symbiotic pair. They co-occur in many tumors, not only just because of the frequency, but they're biased for co-occurring. And this classic paper from Tyler Jackson and Dave Tuvison really tested how synergistic each of these mutations are and what each individual mutation on its own would do to survival in a genetically engineered mouse model. So if we look at a heterozygous P53, that's this survival curve. So it does induce a sort of shortened lifespan due to tumor occurrence. But amazingly, if we lose that second copy of P53, we go down to a very, very short survival curve for these animals. By contrast, if we just have wild type P53, 2 copies, and we add 1 copy of KRAS mutant, we're at this survival curve. So the oncogene gain of function mutation does not do as much as for promotion of cancer as the loss of both copies of the tumor suppressor, which would be here. And then of course, when you synergize them, you get the worst outcome. So this, I think, gives us an idea that in order to treat tumors, we not only need to deal with the oncogenic drivers, but we need something akin to driving more of the profound tumor suppressive activities that might be lost in tumors. So why do we need actually to make a direct KRAS inhibitor? It sounds obvious, but actually when you take a step back and you think, well, where KRAS sits in the pathway, and this is a shortened, abbreviated, highly limited view of the growth factor pathway, starting with KRAS localized at the membrane, you see that its key effector, one of its 2 key effectors is shown here, BRAF. The other one not shown is PI3 kinase. But if we just focus on BRAF, we have outstanding inhibitors of the BRAF kinase. So immediately downstream, we can choke off a tumor that has a KRAS mutant. But we learned in the mid to late 2000s that the BRAF inhibitors showed very, very little efficacy in KRAS mutant tumors. So that really forced us to reconsider our sort of view that even though KRAS was difficult to drug, maybe we didn't need to, because we could go downstream or in some kind of a parallel pathway or look for synthetic lethal interactions. But really, in the late 2000s, we realized that that wouldn't be possible. And so even though we've known about the oncogene since 1983, it's the first human oncogene to be discovered, there had been no approved therapies until last year. And by contrast, BRAF mutations were discovered in 2002, and 9 years later, vemurafenib was raced ahead to full approval. So what's exciting now is that last year, and I'll tell you the story that leads to that, is that there is a direct KRAS inhibitor approved in May of 2021. And why it took so long to do that, how that emerged, will be the first part of the talk, and then I'll go on to the immuno-oncology and the tumor suppressive activities. Told you that it was very quick to develop BRAF inhibitors. That's their ATP competitive nature. The ATP site is eminently druggable. Molecules that compete for ATP are very drug-like, small, 500 molecular weight, have all the ideal properties. But the nucleotide pocket in KRAS, even though it's just a GTP versus ATP, the biochemical affinity for, I'm sorry, let me see here.
Dr. Shaukat, we can no longer hear you.
Okay. Are you there still? I'm sorry.
Yes.
Something popped up on my computer of a recording I did for something else. Okay, am I back? And
yes, if you can just share your screen again so that we can see your presentation, that would be great. Okay, there we go. Yep.
Okay, cool, thank you. So the real challenge for competing GTP out of KRAS is you'll notice that the affinity for GTP for KRAS is in the picomolar range. The other GTPases, RABs, EFTUs, G-alpha-S, heterotrimerics, they have a range of affinities, but KRAS is very, very recalcitrant to releasing GTP. So we would not be able to make a drug-like molecule that could act at the sub-picomolar level and compete for millimolar GTP for the binding site. So with that piece of biochemical information, you might imagine then that the regulation of the GDP to GTP transition for RAS has to be regulated by other proteins. We just couldn't wait for the half-life of the release of GDP to happen an uncatalyzed way. So in order to convert from the GDP to the GTP state, proteins referred to as guanine exchange factors bind to the GDP state of RAS, open up the protein quite dramatically and destabilize the nucleotide, which falls out. And then the cellular high level of GTP relative to GDP then loads back in and therefore produces the active GTP. And I didn't say this, but the two switch regions, which are adjacent to the nucleotide pocket, which move depending on that single phosphate being at the gamma position or absent, then relocalize, reorient these features to make them complementary for high affinity interactions with PI3 kinase and RAF, which I mentioned before, which are the downstream branches of the growth factor pathway. Now, what the cancer cell is going to be selected for in terms of RAS modulation is for a form of RAS that is not subject to conversion of GTP to GDP. And the best way the cell has to do that with the wild type protein is the GTPase activating protein, the GAP, which comes up to the RAS GTP. And shown here, here's the GAP and here's RAS with GTP. It positions an arginine in the vicinity of the nucleotide, greatly accelerating the water catalyzed attack of the gamma phosphate. So what the cancer cell is going to be selected for is a mutation of RAS that will alter the catalytic organization of the GAP-RAS complex. And so it pushes, when you mutate glycine 12 to any other residue, there is a steric clash with the arginine, and the ability of the GAP to catalyze the hydrolysis is vastly diminished by 3 orders of magnitude or more. So that is the single most mutated single residue in all of the oncogenes, and it's this position right at this interface. Now, there is another way for RAS to be converted, and that's through intrinsic hydrolysis, but that's about 4 orders of magnitude less significant, and I'll mention that. It'll come up to be relevant with how the drugs work in a few moments. Now, because there is just a steric mechanism for a gain of cellular function, in other words, breaking the complex between the GAP and the RAS is all you need to leave RAS in the active state. Therefore, we get different kinds of point mutations that give rise to different codons for different amino acids. So the most common mutation at pancreatic cancer is aspartate, then valine, then cysteine. But in colon cancer, cysteine grows, aspartate decreases. And in the smoking-induced lung cancers, cysteine becomes dominant. And so this kind of variability is thought to basically arise by the different carcinogens and that give rise to the mutations. We looked at it according to the chemical features of the residues that are present in the oncogene. And of course, cysteine is a very attractive, unique chemical amino acid. It's rare in the proteome. It's also, importantly, very reactive and highly nucleophilic. So drugs which react with the cysteine can be quite selective for attachment to the cysteine. So even though in the entire group of G12 substitutions across all cancers, which is shown on the right, cysteine is in not the top, that's aspartate, it's not the second, it's valine, it's the third most frequent. Because of its chemical reactivity, we decided to focus on just making an inhibitor of that KRAS G12C. By focusing in that way, and because we had a covalent bond as our goal, we had the opportunity to maybe make an inhibitor that would actually spare the wild type enzyme and not inhibit the wild type. That's the second feature I want to emphasize of going after a covalent strategy, when the covalency is dependent upon the somatic mutation that is present in the tumor. Therefore, you get a lot of more focus inhibition on the tumor cell as long as that target is a tumor cell intrinsic driver, as is KRAS. So we solved the structure, and this was a project done by a postdoc and a graduate student in the lab, Ulf Peters and John Ostrom. John is in Boston in Stuart Schreiber's lab as a postdoc now, finishing up his fellowship. And when they went after this target, we first solved the APO enzyme. You can see in the middle is the glycine 12 to cysteine position. Here's GDP, and here are those two switch regions. And as a mobile protein, some of the elements, amino acids in the switch 2 were disordered and not identifiable in this GDP state. The key thing that we then went after and needed was a drug discovery platform that would get us a first attachment to the cysteine and some kind of complementarity to these pockets here. And you can see already from this picture that when we started, there didn't seem to be any deep pockets that we could engage with. Now, a good friend of mine, a former Dana-Farber faculty member, Nathaniel Gray, worked on exploiting the nucleotide pocket for this same cysteine. And they really developed a lot of heroic chemistry to attach electrophiles to hit the cysteine. And they did achieve that. So this idea of going after the cysteine, I think, attracted a number of people's attention. I think what differentiated our approach is we took a little unbiased approach. And the unbiased way means you have to sort of give up all the starting chemistry chemotypes that you know of and start from scratch. In this case, we used a fragment-based approach called tethering that was developed at a company called Synesis Pharmaceuticals that was founded by my colleague here at UCSF, Jim Wells. And what's really powerful about this approach is that you have a library, an unusual library of disulfide molecules. These disulfides have small fragments. These could be, phenyl rings, biphenyl rings, constructed quite quickly with amides and piperazines, sort of half of a drug, if you will. And what they do is they make disulfide bonds, but then they reverse off, and there's a competitor, beta-mercaptoethanol, in the mix. So there's a lot of back and forth. And so this is what's unusual is it's a thermodynamic-based assay for a covalent reaction, which is quite out there because normally when you do a covalent drug screen, it's a kinetic screen. And once you get the product, you don't go back. But this ability for exchange reactions to happen and a competitor beta-mercaptoethanol, I think was a very, very fortuitous sort of choice for us in retrospect. So once we did the screen, and you do the screen and read it out by whole protein mass spec, so even if they're weak hits, you know the actual molecule is correct. There's very little lack of replication, lack of reproducibility in those early things, which is quite rare for early screens. Usually there's a lot of dropouts later on. And so from the screen, these two molecules, 6H5 and 2E7, emerged that John characterized as binding 100% to the G12C mutant of RAS, but not at all to the wild type. And this protein was in the GDP state. So now let's go back to the nucleotide state issue. If we put the protein in the GDP state, it labels 100%. But if we put it in the GTP state, these same ligands have a hard time, basically don't make a covalent bond. That cysteine is still there, but it must be occluded in the GTP state. So while we liked the subtleness of the change, GDP to GTP, completely changing our molecule reactivity, it went the wrong way. We want a molecule that will label the GTP state, of course, because as I told you, that's the oncogenic protoform of the protein. So we went back to the collection and put the protein in the GTP state, reran the entire screen, and everything came out around this level, which was nothing that we could develop. So we really struggled and had no other options except to say, let's further characterize this binding to the GDP state and hope that there'll be enough cycling of the protein in cancer cells to allow engagement of a GDP selective ligand. But this, I would say, was the biggest sort of red flag in the moment of the project, even though we had a good ligand. Big breakthrough came when Ulf solved the crystal structure of the ligand that John had discovered and showed that it bound in a pocket. And remember, I showed you this apo form of the protein with little small divots. There didn't seem to be a nice pocket, but when the protein co-complex showed up, there was a beautiful pocket with the protein surrounding the drug. The region that surrounded the drug was this switch 2 loop. And that, remember, was disordered in the GDP state. So this is a case where the drug had to be there to show the intermediate state of the pocket being there. So without the drug, we wouldn't have discovered the pocket, if you will. The other feature about the structure was that since we were binding under switch 2, and this switch 2 is half closed, we knew from other crystal structures of the GTP state that this switch 2 was fully closed in the GTP state. So in other words, when GTP is there, this pocket is absent. And remember, I showed you the molecule didn't bind to the GTP state. So all of that made sense once the structure popped out. Now, we wondered, is there any data out there that could tell us whether there's enough cycling of the G12C mutant to have enough of a population in the GDP state so that our drugs could bind and inhibit signaling. And this really nice paper from the Westover lab characterized a number of different mutants of KRAS with respect to gap stimulation. So I told you that when you get a mutant that pushes away the arginine, you drop by about 3 orders of magnitude of the wild type catalyzed gap, and that's true for all of these mutants, some more than others. What was interesting was that the study showed that the G12C mutant has almost wild type-like intrinsic activity. That gave us a lot of hope that there could be enough cycling in order to sort of have a population of the GDP state to go after. Now, since that time, a lot of people have modeled whether these biochemical numbers match what happens in the cell. And there's very good agreement, but there's also some interesting differences. So a lot of studies are emerging to ask, is this the full picture that we need to model? And there are whole sort of proteome-wide screens that Pirolito at Sloan Kettering is doing to understand if other gaps, in fact, can stimulate this hydrolysis. But the bottom line is, it looks like there is enough GDP population for a GDP selective drug to work. And so we were seeing cellular activity, but we knew it was going to need a lot of optimization. This was the molecule we had published on in 2013, and it has a lot of flexible bonds. The protein is very flexible. So we really launched into a company we called Araxes with some former students and colleagues of mine that optimized this molecule substantially. And you can see it kind of gaining a lot more particular kind of substituents, and iodine switched to a cyclopropyl methyl, and the zetadine 4-membered ring to position the acrylamide warhead. And then a big breakthrough came with this molecule, 1620, which pre-organized the middle of the molecule and oriented what was already a good affinity element for reversible binding and the acrylamide at the end. And this pre-organization really matched what the protein's pocket would be most sort of organized as in the apo form. It's hard to always know that, but that's kind of how the data seemed to emerge. And then this was probably about 2016, 17, 18, and then a number of other groups began to make their own versions of this molecule. And the one that has really emerged and was approved last year is sotorasib, but adagrasib is also another leading contender likely to be approved shortly. And you can see they have many of the same elements, the electrophile warhead, the positioning and things, but each of them have a lot of subtleties. Could be a whole talk just about the differences and similarities in recognition. But the key clinical readout was, this was the earliest one reported from the Amgen molecule. And you can see in the dose escalation, all patients had the G12C mutation, and there were very strong responses. Even in some of the lower dose cohorts, the best response emerged. So this looked very, very encouraging. And as I said, last year it led to the approval. And there's a lot of other papers with more full clinical data. So this is really, I think, going back to Tyler's mouse model, akin to patients in this group. Probably about 50% of them have p53 mutations in addition to the KRAS G12C mutation. So if we genetically removed the KRAS mutation in the mouse, we could see this kind of survival curve. If we pharmacologically inhibit the KRAS mutant, but patients have p53 loss of function, then they are only going to sort of benefit by those kind of 6 months, 12 months kinds of extension. And so this is where we began to think longer term, how could we get more up to this curve? And that's what I'll get to at the end. But going back to KRAS, because this is only one KRAS allele that we have an inhibitor of. And of the direct inhibitors that I know of that are in the clinic right now, they all target KRAS G12C. But you see, that's only the third most frequent. Now, there are molecules emerging just late last year that are able to inhibit the other alleles. An important one is a paper in Journal of Medicinal Chemistry from Marathi, where you see a lot of similarities to the G12C inhibitor, but there's no acrylamide. And this nitrogen bears a positive charge, and it makes a salt bridge interaction with the aspartate that gives it some selectivity for G12D, and it's quite a potent molecule. So this is very exciting. It's going towards the clinic. I think it's supposed to go late this year, early next year. And this is a molecule that was published in one of the G12C resistance papers I'll mention in a moment from Ryan Corcoran's lab. That came from Revolution Medicines. And this molecule is a G12C inhibitor. Covalent addition to this alkylamide happens here, but you'll see it looks very odd. And the reason it does is that it's a molecular glue, sort of like Eric's lab has pioneered, where this brings not an E3 ligase to KRAS, but it brings cyclophilin. And so when you bring a large molecule, a protein inside the cell, cyclophilin, up to RAS, you can make a lot more interactions. And that's what's the magic of this molecule. And I think they'll be publishing on that shortly. So those crystal structures will be really fascinating. And our own lab is working more back on the covalent reactions. And so we've got ligands and inhibitors that covalently bind to the serine and the arginine and the aspartate. And we're sort of just moving those forward to the final stages of characterization. So I think this picture is really getting fleshed out, which is great. And we need more and more of these inhibitors for the many patients that have these specific mutations. So as we know in targeted therapy, we get resistance because of the tremendous selection pressure. And so I'll talk about how that's come out. A lot of groups in Boston have contributed to all of our understanding of that, and I'll highlight that. And then I'll get to P53 tumor suppressor. So from Ryan's lab, the first patient that was published and characterized had started with the G12C mutation and was given the adagrasib drug. And after some treatment, became resistant. And the characterization of the mutations that emerged in the patient were mind-boggling. The number of different mutations, both in the binding pocket, this Y96D disrupted the adagrasib binding because it was key to binding of the molecule. It also picked up a mutation at the 12 position to convert the cysteine to phenylalanine. And then on the other allele, there were other 12 mutations and 13, as well as downstream mutations. So what's interesting is that after discontinuation, the G12C mutation stayed around. These other mutations also emerged and persevered. But what didn't happen was we didn't lose the G12C, we just gained other mutations. And that's similar to the other paper here from Dana Farber group in collaboration with Sloan Kettering. And you see that pre-treatment, all G12C, no other mutations, resistance to adagrasib, and produced a whole slew of new mutations. And there were many, many more patients in this study. So we got a good sign that this was how the resistance was going to emerge. But what I want to highlight is that we never lost the G12C mutation. That persevered, that came along. But these other mutations allowed that inhibition of that to sort of lose its efficacy because the cell had other means to drive the same growth factor signaling. So that kind of fed into a project we had been working on quite a bit before, which was the idea that a covalent inhibitor has a number of properties that you don't often have in targeted therapies. They're covalent, so they stay on the protein. They're covalent in this case because it reacts with the somatic cysteine mutation. So it doesn't, that cysteine is not present in any other normal cells. And so we wondered if the drug could inhibit signaling, like I've shown you before and already, could it do other things that would alert the immune system to the cell being a tumor cell by virtue of it having the somatic mutation and the drug attached to the cysteine. So we know that the proteins in the cell are presented processed by antigen processing and presented on class 1 MHC for surveillance by the immune system. But often the somatic mutations in the driver oncogenes are poor at eliciting a good T cell response. But we wondered whether this large drug could be essentially a big enough chemical difference that it could serve as a tumor-specific antigen on the cell surface. So can we take a drug and go into intracellular targets and make them on the cell surface through normal antigen processing? One feature that is kind of neat about this is that we know KRAS inhibits class 1 MHC presentation. And we know that the Amgen group showed that the inhibitor of the KRAS G12C would actually elicit more class 1 MHC presentation. So all of these features seem to go in the right direction for this approach. Now we just need a modality that can sense the presence of this drug on the cell surface. And so we envisioned that the drug would be attached to class 1 MHC to the KRAS peptide. So we took the A201 class 1 MHC and asked if the drug bound to what's shown is the KRAS peptide K5, starting at the lysine 5 position without the drug and with the drug in a cell surface expression assay. You can see that the drug-bound peptide still is bound by A201. In other words, the drug is pointing up, not down into MHC. So it didn't disrupt the normal A201 presentation. So with Charlie Craig's lab, we decided to go after a bispecific T-cell engager, but we needed a drug that would be highly specific for the drug. So we worked with him for the human naive phage library and elicited an antibody called P1A4 that recognizes the drug very selectively and doesn't recognize the KRAS peptide alone. So we can argue and discuss whether that's the right recognition we want, but the first one we got recognized the drug highly selectively, independent of peptide sequence and independently of MHC identity. And Charlie's lab solved the crystal structure, and you can see that the drug is bound deep in the combining site, just as you might expect. The peptide would be floating up here, but in this case, we just solved it, and that resolved part was the drug. So all of that made sense. Now the question is, in cells treated that contain the G12C mutation and you give them the drug, does this antibody specifically recognize the drug presented on the cell surface by MHC? We used a proximity ligation assay to assess whether binding of the drug-specific antibody would be on the MHC with an anti-MHC antibody. And so you can see in 3 different G12C cell lines, drug treatment gives you PLA positivity, but in a non-G12C cell line, with the appropriate MHC that could present, this does not get recognized. So we've got the selectivity and genotype specificity we want. And so now we reoriented the antibody into multiple formats. Importantly, in this one, we fused it to an anti-CD3 and then tried human PBMCs to kill cells. And this is a setting where this original cell is already sensitive to the drug, but we shortened the drug treatment to a point where the drug would go in and label KRAS, but not inhibit signaling enough to die. And that's what's shown here. But then when we add in PBMCs and the correct drug BiTE pair, we can see very good selectivity, and different orientations gave us a different sort of selectivity. So this is the one that we went after. And the key cell line is shown over here. SW1573 is intrinsically resistant to the G12C inhibitors. None of the G12C inhibitors that are known will inhibit the growth of this cell, and that's because the signaling is rewired in a way that makes them essentially resistant to KRAS inhibition of this type of drug. But importantly, when we include the bispecific T-cell engager, we can kill this cell in a drug-dependent way. So we can regain tumor sensitivity when we use the drug as a cell surface tumor marker of the cell. So we're excited about the possibility then of developing a bispecific T-cell engager or another format of these immune oncology biologics to sort of leverage that very first G12C drug from Amgen or the adagrasib one and have patients have more durable responses, because we know from Ryan and the Watt studies that there are G12C mutations still present in the cell. And there might be other ways and other targets that we could think of to serve as tumor-specific antigens. And I mean, my hope is that in the patients that are treated right now with sotorasib and adagrasib, this is already happening, and maybe there are T cells floating around that see that and are contributing to the cell killing. And there are people, there are ongoing anti-PD-1 trials with those agents. Those will be very, very interesting to see, because I think the more we leverage parts of the immune system to kill these KRAS mutant tumors, the better chance we'll have. So that covers the direct KRAS inhibitor side, and I've highlighted the importance of p53 in probably limiting the response of the KRAS inhibitors. And if we want to get to a situation where we pharmacologically take out the KRAS, we won't go much farther up the patient benefit unless we, I think, engage the p53 side. So we are turning our attention to the p53 mutation status. And as you know, p53 is a transcription factor that drives transcription of a number of genes, both pro-apoptotic genes, cell cycle arrest genes. The one I'll focus on is p21, which is a CDK inhibitor of both CDK4/6 and CDK2 cyclin E. And that arrests the cell cycle at G1 and doesn't allow cells to progress until the DNA damage is corrected or cells go into apoptosis. And we've known for a number of years, again, other genetically engineered mouse models, the power of the p53 reactivation can be dramatic. So tumors that have luciferase that are p53 null, if you reactivate their in the TET system for briefly, just even 3 days, the tumor starts to shrink. And then if you go back to the p53 null status, the tumor still shrinks. So exposing a tumor cell that's addicted to the absence of p53 and exposing them to a pulse of p53 can be enough to eradicate the tumor. So in a sense, the bar is even hopefully different than trying to inhibit an oncogene. Hopefully gaining some tumor suppressive activity, even if briefly, could be dramatic. Now, how do we reactivate a tumor suppressor if it's gone? Many tumor suppressors are mutated, deleted, etc., stop codons, but p53 is an outlier, and many, I'm sure, Dan Farber studied this and know it better than I do, but what's amazing is that there are 3/4 of the p53 mutations are missense mutations, and they occur at hotspots, which are at the DNA interface or are stability mutants. And that's different than all the other tumor suppressors like ABC, ATM, and BRCA. There are fractions, but by and large, these are much more severe loss of the protein. And so the p53 here is shown binding to its DNA recognition, DNA element. And when we think about rescuing function, these kinds of mutations that disrupt DNA binding are going to be hard to rescue with a chemical, because we would need a molecular glue that would sit at the interface of p53 and all of the correct DNA recognition elements. That is very, very hard. So we turned our attention to this mutant that other people had also noticed, which is the tyrosine to cysteine mutant, because it's a stability mutant. And our interest was that it was a cysteine, as you might imagine from what we've just talked about. So can we make a molecule that will chemically rescue the stability of p53 and get it to bind and turn on the appropriate p53 response elements? And so I think I'm going to speed up just a little bit so I get done in a couple of minutes. We started, Alan First Lab had been working on this for a long time and used a lot of NMR and design, came up with this carbazole ligand that sits in the Y220C pocket. And we simply said, these are reversible, let's turn them into an irreversible ligand. And so we attached an acrylamide to the carbazole, and we got good cellular engagement as read out by this click probe. So we already had, a great lead from Alan First Lab and converted it into a covalent probe. And when we solved the crystal structure and we compared our covalent structure with his other ligand, we saw that our molecule flipped around. So we had, and I won't be able to go through all of this, but we had a lot of, when we're trying to rescue function, we're not just trying to make the covalent bond and jam a wrench in the engine. We have to kind of reorganize it, but in the correct way to be more stable. And so that involved a lot of looking at other interactions in the pocket. And at some point, we realized the carbazole was just too big to add different substituents. So we chopped off one of the phenyl rings and turned it into an indole. And at this stage, we had an aldehyde and various substituents off the indole and a methyl acrylamide. And the labeling kept increasing and the crystal structures kept getting more interesting, making new interactions. And the last molecules here really show the emergence of a much, much more ideal molecule. So we took the indole and made a pyridine substitution, which was one big breakthrough. And then we made piperazines and pyrazines and then fused pyrazines. And what you can see is the thermostability of the mutant kept increasing. This 8 degrees is kind of a magic number for us because when you compare the wild type to the the wild type in light blue to the Y220C in dark blue, that's about an 8-degree shift. But if we put these last two ligands on, we can shift all the way back to wild type. And the way we did that is we created a new interaction with this aspartate that moved a loop on P53 into a new orientation that is not present in any other P53 structure. So in order to fix the loss of this tyrosine, we needed to make new interactions and sort of bend the protein in slightly new ways, or at least that's what the chemistry ended up doing. And that's our sort of hypothesis. And what's very nice now with these molecules is that in syngeneic model systems, we can now, so here's the control readout, GAPDH. Here's P27, CDKN1A, MDM2, of course, is the E3 ligase. And you can see that when we add our ligand to the Y220C, we get tenfold and more induction of the P27 gene, just like when wild type is there and you inhibit the E3 ligase, you can boost that. But the nutlin does not work in the mutant setting, but our ligand does not work in the wild type setting. So again, we have selectivity. Here, patient-derived cells, one is Y220C and one is wild type. And again, the specific Y220C activator works. Importantly, we shifted these cells to grow at 28 degrees for an hour while we treated the drug, and then washed everything away and shifted to 37. This gave us sort of the folded state of the protein for the drug to bind to. But when we shift back, then the drug is gone and the drug is all locked in. So all of that is sort of very up to the moment of where we are with this. So the last slide just sort of highlights that there's been tremendous amount of basic discovery on RAS and P53 starting from the late 70s. And now in the last few years, we see now emerging inhibitors of RAS, now the first one approved. P53, there's a company called PNV Pharma that has a Y220C ligand, again, non-covalent, that's just entered the clinic last year. So even P53 mutants are being drugged in the way I described. Now, what's going to be great is to see these two fields come back together in the drug discovery space and see if we can get patients to really jump to tremendous clinical benefit. But that's going to be, I think this year will be a very, very exciting year for both RAS and P53. So I'll look forward to hearing about that more and telling you more about it in the future. And so, so the G12C work was done by people I mentioned, John Ostrom and Ulf Peters. Another number of other people in the lab in the past have worked on various other aspects of drugging other KRAS alleles, and other current people in the lab that I didn't get a chance to tell you about their work are working on the other alleles. And then Jim provided the thinking behind the tethering library, and Araxes optimized the ligand to those clinical molecules. G12C immune targeting was by postdoc Xiang Zhang with a graduate student Peter Reuter in Charlie Craig's lab and Giselle Knudsen in Charlie's lab. And then the activation of P53 was by a postdoc in the lab, current postdoc, Keelan Guiley, who's working on the Y220C as well as other mutants. And on the bottom are people who funded the work. Thank you very much.
I should unmute myself. Thanks, Kevin. This was fantastic. While there are more questions trickling in, let me start with one. The concept of presentation of covalently modified peptides, one could imagine that's more common. That's not just the KRAS. And the question is, have you or anyone else looked in the context of KRAS specifically or more broadly, whether they're naturally emerging either TCR species or even antibodies that emerge against those drugs or peptide conjugated drugs?
Yeah, we're trying right now with Drew Pardal's lab at Johns Hopkins to do exactly that. Sort of, they have these great co-culture systems and single cell sequencing where you can look for TCRs that might have emerged. And we're not doing the clinical samples, but I think what you're getting at is if somebody should do the clinical samples and analyze the TCRs, I hope that that's going on. We're doing it sort of the de novo side. Yeah.
Then we have starting to have some questions here in the Q&A. The first one, could these covalent modifications enhance MHC2 related responses as well?
I think so. I just don't know enough about, I think those peptides with, of course, a little bit longer would be presented by class 2, but I've never been able to wrap my head around how the class 2 contributes to the cytotoxic T cell response. So it just kind of is a little confusing to me. Yeah, probably if I knew the immunology better.
Yeah. Then the next question also along the lines of the immunologic response. Are there CD8 T cell epitopes in G12C, D, and V neoantigens?
Mm-hmm. Yeah, you mean is the G12 containing peptide from KRAS a no neoantigen? Yes. Again, Bert Vogelstein and Drew Pardal's labs have actually made what they call nanobodies, which are mutant-specific antibodies that recognize the G12D and V, I believe, when it's presented on, and I believe it's A201. So they are epitopes. Yeah.
I should probably acknowledge who asked those questions. So the first question was from Chelsea, and the second question was from Rami Reddy. Then we have another question from Ijung Kim. Is G13C also targetable as it has cysteine?
Excellent question, and shockingly to me, no. With the molecules I showed you, we spent a long time trying to make the electrophile a little bit longer, but when the cysteine is kind of pointing out of the protein, there's really, it doesn't seem to be able to be targeted with the switch 2 pocket binders. And it's actually because of the co-neighboring lysine 16, which helps position the acrylamide. So now when you're just literally one position away, you drop all reactivity. There is a molecular glue that hits G13C, but that's again using cyclophilin. So it uses a different modality. But oh boy, I wish the answer to what you asked was yes, because we would have saved a lot of time.
Then we have a question from Bart Latterbach. Nature has created a very high affinity ligand for RAS and GDP. Do you think there is potential to gain higher affinity with current libraries in chemistry? Has there been screens for competitive inhibitors that failed?
It's a great question. It seems like the hard part is that RAS, when it doesn't have the nucleotide bound, it just falls apart. So if you could somehow hold it there and then leverage and start getting a good starting point, you're right. I think picomolar affinity isn't the biggest problem. That is overcomable, but it's hard to get the protein in a way to screen it. The other feature that's bad is that a lot of the interactions that are really affinity driving are with the phosphates. So any ligand you would find that would get that kind of high affinity is probably going to have negative charge, and then is going to be a poor drug-like molecule. So it's kind of you keep running into roadblocks that I see with that strategy. I keep thinking the way you have. But.
Yeah. I think Nathaniel spent a lot of time trying to get around the phosphates.
Yeah, exactly. And he said just it gets so hard to,
It's the same problem with phosphatases. There are many targets where the nature of the pocket defines the chemistry of interaction.
That's right.
Then I have a question here in the chat that I'm going to take from Binchu Liu. Thank you for the super great talk, really exciting. Do you know how many MHC1 haplotypes can bind G12C peptide? If not all, will you think there's a possibility that a covalent molecular, I assume glue, can enhance G12C binding to more broad MHC haplotypes?
Great question. I believe it's, if you just kind of looked at the top quartile of the most frequent MHCs, if you just look at those, there are a subset that can bind, but not that many. I forget the number. It's probably less than 6 or 10 that have sufficient affinity. And of course, we got to predict whether the mutation is pointing up towards the T cell receptor so it could see it as non-self. But I think the last part of your question was about whether the peptide with the drug on it could actually then bind to a different MHC that the parent peptide didn't. And that is a great idea. And we're working on that exact thing for essentially molecular glues that bring down KRAS peptides to MHCs that do not normally present them. And it's looking like that's possible for sure. Yeah.
Thank you. I think we covered the questions and we're also pretty much at time. So thanks, Kevan, again. This was fantastic.
Thanks, Aaron.
Hope next time we get you out here in person.
Yeah. Very close to that for sure.
Thank you.
Stay well, everybody.
Bye.